So my name is Arturo Sala, without a final H. Otherwise, I would play for Liverpool and be millionaire. Okay, so just a brief introduction of the work in my lab. So we work on rare cancers and rare genetic diseases, and the main lines of research in the lab are Friedreich's ataxia, neuroblastoma, and adenoid cystic carcinoma, which is the subject of my talk today. So, just a few words on adenoid cystic carcinoma, or ACC. It is a malignant tumor affecting exocrine glands, mainly salivary glands of the head and neck, but can also strike organs like the breast, the skin, and other exocrine glands in different body locations. In terms of histopathology, ACC is divided mainly into three types. Cribriform and tubular, and as you can see from these microscopy slides, it's a differentiated type of tumor. It's not too dissimilar from normal salivary glands, whereas the solid type of ACC is undifferentiated and is more aggressive, the most aggressive subtype of ACC. It is important to mention that there is no standard systemic chemotherapy for ACC. It is a slow-growing tumor, but relentless, and resistance to chemo and radiotherapy is a problem. About 40% of ACC patients develop metastatic disease, and these patients do very poorly. And this is summarized by this survival curve. In this particular case, it's a survival curve of patients with ACC of the lungs. And the non-operable group succumbed to the disease very quickly, within 5 years, whereas the operable group do better, clearly, but in the long run, they will also die of the disease. So ACC is a rare type of cancer. There are about 2,500 cases in Europe. However, the socio-economical impact is relevant. It mainly affects people in a relatively young age, so in working age, and it mostly affects women. And the tumor can leave permanent disfigurement after following surgical treatment. So a major molecular change found in the majority of ACCs is the alteration of a gene called MYB. MYB encodes for a protein which is highly conserved during evolution. So it's a very important protein, essential for life. However, in some cases, this protein gets changed, altered, like in ACC, and becomes oncogenic. For example, in viruses, there is a class of viruses that infects birds that carry a copy of the MYB gene that is oncogenic, and indeed this MYB, this avian MYB, causes leukemia in birds. Now, our collaborator Goran Stenman published a seminal paper a few years ago in which he was the first to establish that MYB is indeed very, very important in ACC. So he observed that two chromosomes get fused together, leading to the activation or alteration of MYB. And this is maybe a complicated figure, just to illustrate for those of you who understand chromosomes, that there is a fusion between chromosomes 6 and 9, and this leads to the formation of a so-called chimeric gene, so a fusion gene that connects MYB to another gene called NFIB. Now, it is not clear whether it's MYB itself or the fusion gene that is leading to the cancer. And this is something we are actively trying to investigate. Now, in a study funded by an American charity called ACCRF, or Adenoid Cystic Carcinoma Research Foundation, in collaboration with Goran Stenman, we have identified a kinase that is a protein that is able to change the activity of other proteins in cells, called ATR. ATR is activated by MYB in gland cells. So this finding is important because ATR is an enzyme, and therefore it is potentially it's potentially inhibited by small molecules. So we hope to use these small molecule inhibitors as a way of targeting ATR in ACC. This is another maybe complicated figure, but just to summarize the results of our research, we found that both MYB and the fusion variant of MYB, called MYB-ATR, are able to activate a common set of genes that are mainly involved with cell proliferation or DNA repair. And we think this is very relevant because among these genes there is this kinase called ATR that I was describing before. Now, for those of you interested in complicated signaling pathways, ATR as well as ATM are kinases that are activated by DNA damage, and this sort of DNA damage signaling ultimately leads to growth arrest. So the cells will stop to proliferate in order to repair DNA. Now, what we found is that ATR and MYB are linked in ACC tumors. So when MYB expression is high, ATR expression is also high, and we think we can use this knowledge to target ATR using drugs. Just another slides taken from our paper in which we show that MYB as well as MYB and FIB, so the two variants of MYB, are able to transform, so to render gland cells tumorigenic in this in vitro assay. Now, so how ATR and MYB are connected? I'll try to explain. So after the chromosomal translocation leads to the activation of MYB or MYB and FIB, these proteins that are able to actually change gene expression, so they will sit on regulatory sequences near genes important for cell cycle progression or drug and radiation resistance, such as ATR, ATM, and BAB1. And this leads to a more aggressive type of cancer. So, ATR potentially is an excellent target for therapy. So we want to use drugs to inhibit ATR, and not MYB directly, because MYB is difficult to inhibit, and hopefully leading to a cure in the future, at least for this cancer. So there are small molecule inhibitors already available that block ATR activity. One is an inhibitor called VX970, also known as M6620. It is a clinical inhibitor, has been used in phase 1 and 2 clinical trials for a number of solid tumors in combination with radio and chemotherapy. So we have done a pilot study in which we have used this inhibitor in a mouse model in which a patient tumor was growing. So it's been a patient tumor has been transplanted into a mouse, and then we have tested the VX inhibitor in this system. And to cut a long story short, we found that the ATR inhibitor on its own was able to inhibit the tumor growth for 64%. Clearly, the tumor was expressing high levels of ATR and its activated form called phospho-ATR. So these results, I think, are promising also in the view of this. This actually is a single mouse from that experiment showing that the tumor actually regressed in the presence of the drug, and this is something that is very rarely observed in this type of tumors. So what we want to do with the Oracle Cancer Trust support, we would like to validate this MYB-ATR axis using patient-derived cells, and we want to use the inhibitor, the ATR inhibitor, actually multiple inhibitors, because there are a couple of clinical inhibitors for ATR, and use this in combination with radio and chemotherapy in mouse models and patient cell lines. And this will be facilitated by our collaboration with Professor Göran Stenman in Gothenburg, which holds all these patient models and also will provide expertise with the pathological analysis. So we predict that the small molecule inhibitors in combination with chemoradiotherapy should sensitize the tumors and potentially offer new hope for patients with this terrible type of cancer. And I'd like to finish here by acknowledging the members of my lab, and a PhD student, Ylenia, has been just recruited to carry out this project, the Oracle project. But I have also other people working on neuroblastoma or Friedreich ataxia. And I would like to thank the collaborators, Göran Stenman, Matthias Andersson, on this ACC project. And of course, funding by Oracle and also other charities and the MRC that fund my neuroblastoma and other Friedreich ataxia research. And I'm happy to take questions. Thank you.